Hey there. So I know staring down a massive molecular biology exam can feel, well, completely overwhelming. But don't sweat it. Think of me as your peer tutor, and together we are going to absolutely conquer this material using this highly structured explainer. We're going to break down the complexities of the cell, map out all the tiny molecular machinery, and make sure you walk into test day with total confidence. Let's do this. Here's our quick syllabus for this explainer so you know exactly what's coming. 1. Central dogma and glossary. 2. The cell cycle S phase. 3. Unzipping the fork. 4. Polymerase all-stars. 5. Prokaryotes versus eukaryotes. And finally, 6. Transcription and code. Okay, let's dive right in. Kicking things off with Section 1, Central Dogma and Glossary. So if there is one absolute unbreakable rule in molecular biology, it's Francis Crick's central dogma. It's basically the ultimate flow of information in nature. Information is securely stored in your DNA, it gets transcribed into RNA, and then that RNA is translated into proteins. And those proteins, they go on to do almost all of the heavy lifting in your cells. Honestly, understanding this directional flow is the absolute key to everything else we're going to cover today.
First up, let's nail down a crucial distinction: genotype versus phenotype. Your genotype is your complete genetic blueprint. That's the whole shebang, including both the coding genes that make messenger RNA and the non-coding genes that make things like transfer RNA. Your phenotype, on the other hand, is what actually gets built from that blueprint. It's the set of physical, observable characteristics you can actually see.
Okay, so how do we talk about the variations in those genes? Well, a gene sits at a very specific spot on a chromosome called a locus, and the different variant forms of that gene are called alleles. If you inherit two identical alleles at a specific locus, like say two identical genes for blue eye color, you're a homozygote. But if you inherit two different alleles, maybe one for blue eyes and one for brown eyes, you are a heterozygote. Keep these structural definitions locked in your memory bank.
Moving right along to Section 2, the cell cycle S phase.
Let's look at this simple logic flow of the cell cycle. A cell actually spends most of its life just hanging out in interphase, which is broken down into G1, S, and G2. But the absolute critical moment for us and for your exam is that S phase, which stands for synthesis. You see, the main rule for cell division is that the DNA has to be copied first. The S phase is that exact specific window where the cell strictly replicates its entire genome, creating identical DNA strands so that when it finally hits mitosis, both of the new daughter cells get a perfect, complete copy.
All right, let's drop down to the factory floor. Section 3, Unzipping the Replication Fork. So, picture this. You've got the enzyme Helicase acting like a wedge, doing the really hard work of breaking those weak hydrogen bonds to separate the two DNA strands. But as you pull apart tightly twisted strands, the DNA ahead of the fork gets super tightly coiled. Enter topoisomerase. It acts like a stress relief massage therapist, attaching to the fork to relieve that massive twisting tension so the DNA doesn't just snap.
Let's put this all together to see how our semi-conservative replication journey works. Semi-conservative is just a fancy way of saying the new DNA consists of one old parental strand and one newly synthesized strand. To make that happen, helicase breaks the bonds. But wait, what stops the DNA from just snapping right back together? Single-strand binding proteins, or SSBs for short. They attach to the separated strands and strictly prevent that DNA zipper from reattaching, all while topoisomerase manages the stress up ahead.
Honestly, a great way to think of the replication fork is as a literal molecular zipper getting unzipped. As helicase just plows right through, all the molecular machinery moves in, and free nucleotides floating around wait to be matched up perfectly to the newly exposed original template strands.
All right, rapid-fire quiz time to help you dodge a classic exam trap. Why is the 3' end absolutely critical for DNA polymerase? Well, the answer is that DNA polymerase is incredibly strict. It can only add new nucleotides to the 3' end of a DNA strand. It literally cannot start a chain from scratch. Think of the 3' end as the absolute only functional docking station where new nucleotide cargo can be attached. Because of this, the new strand always has to be built in a 5' to 3' direction.
Now, what's really wild is that this strict 3' rule creates a massive mechanical headache. Because the two DNA strands run in opposite directions, only one strand can be built continuously as the zipper opens, and we call that the leading strand. But the other side, the lagging strand, has to be built discontinuously, moving backwards away from the fork. It's built in these choppy, short segments called Okazaki fragments, which later have to be glued all together by an enzyme called DNA ligase.
Next up, Section 4, the DNA polymerase all-stars. So eukaryotic cells actually rely on a whole team of 5 main DNA polymerases: alpha, beta, gamma, delta, and epsilon. 4 of them live right there in the nucleus handling standard cellular duties. But one of them is a total specialist located entirely outside the nucleus. You're definitely going to want to memorize this summary of their functions for the test.
Now here is a step-by-step memory strategy to make absolutely sure you nail these on the test. Ready? First, think gamma for genetics of the mitochondria. Polymerase gamma is solely responsible for replicating mitochondrial DNA. Next, think beta for better fix that damaged DNA. Polymerase beta functions primarily in the excision repair of everyday DNA damage. Finally, alpha, delta, and epsilon are your active dividers in the nucleus, putting in all the heavy lifting during replication.
Let's shift gears slightly. Section 5, Prokaryotic versus Eukaryotic Replication. So how do bacteria handle this compared to us? Well, prokaryotes, like bacteria, have small circular genomes. Because of that, they only need one origin of replication, just a single bubble that grows and grows until the whole circle is copied. But eukaryotes, like us, we have massive linear chromosomes. If we only had one replication bubble starting at one end, it would literally take hours upon hours to copy just one single chromosome.
I really want to hammer home this common exam pitfall. The absolute fundamental difference here comes down to sheer necessity. Eukaryotes desperately require many replication bubbles firing off simultaneously across the chromosome. Without those multiple origins of replication, efficient genome copying would be impossible, and complex cell survival would simply fail.
And finally, we've reached the last leg of our cram session. Section 6, Transcription and Genetic Code.
Let's jump back to the central dogma for a second to look at transcription. This process happens right in the nucleus. RNA polymerase binds to a specific DNA sequence called a promoter, and it transcribes a single strand of messenger RNA. But before that mRNA can actually be used, it goes through some crucial processing. The non-coding segments, which are called introns, are spliced out, leaving only the coding exons. Then, protective caps and tails are added to the ends to keep it safe.
Once that newly processed mature mRNA hits the ribosome, it's read in little 3-letter chunks called codons. You absolutely have to know 3 key characteristics of this genetic code. 1, it is universal. Pretty much all living organisms use this exact same code. 2, it's unambiguous. A specific codon will only ever code for one specific amino acid. And 3, it is redundant, which is also known as degenerate. This just means most amino acids are encoded by more than one codon.
Now you might be wondering, wait, why would a redundant degenerate code actually be a good thing? Well, because mistakes happen. Everyday chemicals and UV radiation are constantly damaging our cells. This redundancy acts as a massive evolutionary advantage. It's basically a built-in safety buffer for everyday mutations. If a nucleotide changes due to a mutation, the new mutated codon might actually still code for the exact same amino acid, which means the resulting protein functions perfectly fine, despite the DNA damage. Pretty cool, right?
But I want to leave you with one final thought-provoking question before we wrap up this explainer. What if a mutation actually bypasses that safety buffer? If a mutation changes the nucleotide sequence and the new codon codes for a completely different amino acid entirely, what effect will that single alteration have on the 3-dimensional structure and the ultimate function of the resulting polypeptide? Think about how structure dictates function in biology. Keep that core concept in mind, study hard, and you are going to absolutely crush this exam.